Can I ask you what this key is here? What?
Oh yeah, this key, yeah, that's a key that when I was an undergrad in Trinity College, my third year I joined the subject of genetics, and there was a building for genetics at the time, a really old tumbling down building that I think was originally Oscar Wilde's dad's hospital for doing operations in. So it was actually quite
Was Oscar Wilde's dad a surgeon?
Yeah, he was a big Sir William Wilde.
God,
he was
I
a
didn't
big
know that.
eye surgeon and stuff. So, but anyway, so the building's been knocked down since and replaced with a much more flashy institute. But at the time when I joined it, there was a guy, George Dawson, who was the head of the department. And the very first thing when we were just this kind of little green third year coming into the next department, he'd give every one of us a key to the building. And this is a building full of like expensive equipment and stuff. So it was kind of like, it was just like suddenly being trusted with something.
Absolutely. And what age would you have been?
I don't know, what's the third year? Would that be about? Be probably about 20 or
20,
something
yeah.
like that. Yeah.
Amazing. You might tell us maybe what your name is and who you are.
Oh yeah. I'm Denis Shields. I'm based in the Conway Institute in University College Dublin, and I work in the field of what's called bioinformatics, and it's basically studying DNA and protein sequences on computers and looking for patterns. So
you're not in a lab with a pipette?
Yeah, no, not really. I started out when I was an undergrad. I tried doing lab work and I just didn't have the patience for it. You have to have a certain mentality to be good at lab work, and I wasn't really good at it. So I I ended up on computers, and so I do a lot of computer analysis. But what this came out of was there was a call for grant ideas about COVID, and when COVID came out first, everyone was trying to think, oh, it's really not very nice. I should be doing something too. So I kind of said, well, what should I do? So I kind of thought, well, I know a bit about peptides, which are small bits of proteins. We'd done some experiments in the past in heart disease, trying to look at them. And so I thought, well, can I apply some of that, what I learned from that, to designing peptides against COVID? And I was saying, how am I going to do it? And I was thinking about different bits. But I can't even understand this picture now. It makes no sense to me really. Well, it makes sort of sense. I think if I spent half an hour, I'd probably, but at the time it was useful for me to get my ideas straight, just scribbling all this down. So
you were kind of thinking immediately, and there's some kind of codes and different, you understood, if you looked at it again properly, you might be able to.
Look now, and I'm going to explain it all to you in a lot of detail. Yes, there's different, peptides. So there could be like little motifs in peptides. There's a famous motif on our blood cells, our platelet cells in our blood, called RGD, which is important for binding. It's on the fibrinogen, the clotting protein that binds to these platelet blood cells. So that's a very important motif, and I was interested, could you build from known little short motifs? So RGD just means 3 different building blocks of the amino acids that make up protein.
Yeah.
And then there's other building blocks, and I was trying to think, well, what if we made combinations of these, put them together in ways that we could maybe make them only be activated when the virus is infecting the cells. So basically I thought about this and thought, well, I can do some computer analysis to try and come up with these things, and what would be interesting parts of the viral protein to introduce into these peptides along with other bits of peptides. And then I found collaborators in the protein chemistry, the peptide chemistry, people who can make these things, and in virology, so Virginie in virology, and Dave O'Connell in the protein biochemistry end of things, to help us make some more of these peptides. So we've kind of working together on that. So this initial squiggle diddly has led to a grant where we're actually just coming to the end now this summer of some of the work on that. So.
Okay. So when you say you identified these motifs, did you kind of just dream them up out of your head, or?
Yeah, a lot of them, I think, in retrospect, probably what we did in the grant, what I said I was going to do in the grant, are a little bit different to what we probably ended up focusing on when you actually sit down and start doing things. But some of these were based on motifs that are in the virus sequence that we know bind. Like there's a motif called RGD. It's actually in the coronavirus spike protein. So that RGD, that was why it was interesting as well, apart from what I was saying about it being important in blood clotting cells. So, and then this one, the NRP1 binding motif, is just after a cleavage site where the spike protein gets chopped off. It reveals this binding site for this protein called NRP1. So I thought, could we make something that combines some of these motifs together? Would that have interesting signaling properties? That was my original thought, and we're kind of doing that at the moment. I think we've shifted our direction a little bit away from those 2 motifs for a number of reasons, but the principles of the sort of the idea behind it are there.
Okay, so probably your background in genetics and has allowed you to really fully understand the genome, the code, and then be able to, like when you see the like letters of the code, can you then sort of almost visualize the protein or?
Yeah, sometimes it's easier than others. I mean, sometimes you can visualize little aspects of the protein, but not the whole thing. There is great software now that's just come out the last 2 years called AlphaFold that uses machine learning that actually works, that is very good at predicting from the sequence of letters the way that string of a protein kind of folds up into a three-dimensional thing, a particular shape. And so that there is good software out there that does that. But sometimes I'm often just looking for simpler patterns, actually, to be honest. And sometimes it's things like, it's understanding the evolution of these things. So if we compare the SARS-CoV-2 with maybe the common cold coronavirus, and then you look at the spike protein and you say, what bits are different here? Does that give clues? Is that the bits that are the same, are they important, or are the bits that are different because that's what makes a difference between them?
So this is an interesting thing as well. This is a piece of paper that I'm looking at here that's just covered in all kinds of numbers and letters and sequences. So just tell us briefly what that is.
Yeah, what this came out of was a few years ago I joined a music group that plays Indonesian music. So there's a very talented musician based here in UCD and also in the concert hall, Peter Moran, and he leads a group. And when I heard them play, I just thought it was the best thing I'd heard in a long time. So I ended up joining the group, and so we've played at a number of interesting venues, and it's a very interesting music because it's in a totally different parallel universe to Western music. You've got your different scales and different structures and a whole different philosophy and mentality about the music. So it's just great fun. And then I was just sort of sitting at home one night and I thought, wouldn't it be really crazy to try and design a piece of music for this orchestra based on something from DNA or protein? And when I thought about it, I thought, well, the great thing about collagen is a protein which it has kind of triplet repeats in it. And I thought, well, triplet repeats sounds like music to me. So, and it also, like, then the DNA code itself is a triplet repeat. So you've got a triplet repeat within a triplet repeat, which is sort of like, I thought I'll have to play with that. So I looked for a chunk of collagen that looked as though it was repetitive enough and might have a bit of structure to it. So I just took a bit out of, there's a collagen 1A1, one of the big collagens, really important to our body. Like about 18% of our protein in our body is collagens. So they're important group of proteins. So then I just tried to assign notes to certain amino acids. Now the scale only has 7 notes. Yeah. So and we have 20 amino acids, so I had to collapse it down a little bit, which is fine. I just, amino acids that are similar, I just gave the same note to. Exactly. Yeah.
What is this actually?
That's a power pack for a computer, yeah. So that's just, it doesn't do any of the thinking. All it does is just passing electricity, and it's got a fan in it. And I don't really know a whole lot about how computers actually work. I've never actually taken one apart, but like most people, I use them. So.